Did you know that smoking can cause diseases such as lung cancer? Yeah. Do you know why smoking causes cancer? No. Well, it's because smoking can lead to DNA damage. What? Let me explain it to you.
Cigarettes contain over 4,000 chemicals, including arsenic, formaldehyde, and methanol. These chemicals are carcinogenic, meaning they cause cancer and contribute to DNA damage. DNA is made up of 4 nucleotides: cytosine, guanine, adenine, and thymine. Together, these nucleotides form a code. Each piece of the code, or gene, tells cells to make specific proteins. Not only does smoking cause damage to individual DNA strands, it can also cause DNA methylation. DNA methylation is the process by which methyl groups, or CH3, attach themselves to a cytosine group of DNA, becoming a 5-methylcytosine group. The methyl group physically blocks the code from being read during transcription. Because the code is blocked, the protein this gene codes for cannot be made. Methyl groups do not alter the genetic code of DNA, but simply work like a light switch and turn genes on or off.
DNA methylation has been connected to cancer because sometimes methyl groups attach themselves to genes that control cell growth and division, like proto-oncogenes.
Proto-oncogenes produce proteins that stimulate cell division. When mutated or methylated, proto-oncogenes become oncogenes, which cause cells to become cancerous and divide out of control. If a methyl group turns off a proto-oncogene, turning it into an oncogene, then the gene will no longer mediate cell growth and division, causing the cells to grow at a rapid rate. Oncogenes cannot produce the proteins that tell cells to stop dividing, so cells will continue to divide. Methyl groups can also silence tumor suppressor genes, which are genes that slow down cell division, signal to cells to die, and repair DNA damage. The silencing of these genes will make a person more susceptible to cancer, as the genes can no longer slow down cell division. It is important to know how DNA methylation affects the genome, and more importantly, the epigenome. The genome is the compilation of all the genes in the body, while the epigenome consists of groups of chemical compounds that determine how the genome is expressed. When methyl groups are removed or bonded to genes, they mark the genes as either on or off, and by doing so, change the way the individual genes are expressed. Your genome is like the script to the play of your genetics, and methyl groups determine which lines get read. Lifestyle choices, like smoking, affect changes in the epigenome. DNA methylation from smoking can have lasting effects on the epigenome. A recent study found that methylation patterns attributed to smoking were still present on the DNA of people who had quit smoking 30 years earlier. This DNA methylation doesn't just affect smokers either. Babies whose mothers smoked while pregnant were found to have a 2.3% increase of DNA methylation on the AXL gene. The AXL gene is a gene that codes for a protein that controls cell functions including growth and division. An increased methylation of this gene can lead to cancer.
Seems like I'm just doomed, huh? Actually, the sooner you stop smoking, the faster your DNA can repair itself. And the longer you smoke, the more your DNA is damaged. Well, you've convinced me. Oh my goodness, do you need me to explain the impact of littering too?